Okay, the next technology what we are going to study in biotechnology is gene therapy. First, we should know what is the meaning of therapy. It is a medical term. Therapy means treatment. Therapy means treatment. For example, we are having malaria fever. We are suffering. Then we are going to doctor. The doctor is giving treatment to make the malarial fever to cure. It is called a treatment. Doctor is giving us treatment. The treatment is nothing but therapy. Suppose we have some problem in the joints or problem in the knees or anywhere in the body. We are going to the ortho doctors, bone specialists. With the help of physiotherapists, they are giving physical exercise to make that problem in the joints to become alright. It is called a therapy, physiotherapy. Physio means physical exercise, therapy means treatment. So the treatment is called a therapy. Okay, if it is done with the help of chemicals, that is medicine, chemotherapy. If it is done, the treatment is done with the help of physical exercise, it is called a physiotherapy. Now in biotechnology, there is another one therapy which is called a gene therapy. Now we are having so many diseases which are caused by microorganisms. They are called infective diseases. Okay, infectious diseases we can call. That is maybe bacterial cells or viruses or the fungal parasites, anything they can enter into the body. They can cause varieties of diseases. They are called infectious. There are certain diseases in our body, non-infectious, like a heart problem, lungs problem, okay, kidney disorder, okay, all these things we can call it as non-communicative, non-infectious. That means these diseases cannot spread from one person to another. The third type of diseases are called genetic diseases. We have a lot of body cells. In each and every cell there is a nucleus. Inside the nucleus 46 chromosomes are present. In the 46 chromosomes, enormous number of paired genes are present. The genes are expressing various characters in our body. The genes are controlling all the activities of our body. The genes are regulating the production of enzymes. The genes are regulating the growth of the body. The genes are regulating the production of hormones in the endocrine glands. Like there are so many things. They are controlling. So genes are important. The genes are nothing but DNA molecules. Okay, this is the outline. Assume that one person is having genetic defect, genetic disease. Okay, then how to cure that genetic disease? The genetic disease, okay, genetic disease, one disease, any one disease we can take. The genetic disease is caused due to, it is caused due to defective gene, defective genes present in the chromosomes. Okay, present in the chromosomes. Okay, I will give one example. The diabetes is a genetic disease. If father is affected by diabetes, the children will be affected. If the mother is affected by diabetes or grandparents are affected by diabetes, the diabetes is affecting the next generation. Generation after generation, the problem is there. It is due to the defective gene. Okay, it is due to defective gene. Just before, in the previous topic, we studied that the pancreas is having two types of cells, the islets of Langerhans, alpha cells and beta cells. Now this is beta cell, okay, one beta cell. In the beta cell, nucleus is there. In the chromosome, one gene is there. Now assume that the gene is normal gene. Okay, the gene is a normal gene. Normal gene. This gene is for the production of insulin. So normal amount of insulin will be produced. But due to some problem, imagine that the normal gene is changed into a defective gene in one particular individual, in a particular generation. It is due to mutation, sudden change in a gene, mutation, defective gene. The defective gene cannot produce, okay, insulin. The defective gene cannot produce insulin. If the insulin is genes are not produced sufficiently, the person will be affected by diabetes. So the diabetes, the gene, defective gene will pass on to the subsequent generation. So all the individuals will be affected by the diabetes. This is a common thing. What is that? It is a genetic disease. Now the diabetes is called a genetic disease. Now how to rectify this problem? We can rectify this problem with the help of gene therapy. Now what we have to do is, we have to take the diabetic patient and we have to take the beta cells present in the pancreas. Beta cells present in the pancreas. Okay, now I am giving only the meaning of a gene therapy. Now these are all the islets of Lagrange, the patches. Now here the beta cells are there. Okay, this is one beta cell. Here one defective gene is there. In all the beta cells, defective gene is there. So that the defective gene is not producing insulin. Diabetes is caused. Now what we have to do is, in one cell, not in all the cells, one cell you have to take, you take this defective gene away. Okay, defective gene should be removed. It should be removed. And what we have to do is, this is a normal gene in the beta cells from other individual, normal individual we are taking. This is a normal insulin gene. Insulin gene taken from normal individual. Taken from normal individual. We have to take it. And just we have to introduce the gene in that case, because defective gene is removed. The space is there, we have to bring and fit in the normal gene. So once the normal gene is there, this cell is perfectly normal. This cell should be allowed to multiply in this islets of Lagrange. So like that, in all the islets of Lagrange, one cell we have to make, correct? So once the insulin genes are becoming normal, more in number, then the insulin production again it will be started. Then the individual is slowly becoming a normal individual with a normal amount of insulin. This is called the gene therapy. Now we take what is the meaning of gene therapy. It is the corrective treatment, or it is the corrective measure. Corrective means correcting defective gene into normal gene. Correcting the defective gene into normal. That is corrective gene therapy. Okay, it is called the corrective gene therapy. So in this treatment, the defective gene is corrected into a normal gene. Okay, that is called the corrective gene therapy. That is called the gene therapy. Meaning for that, the explanation is given. But in the gene therapy, there are two types of gene therapy. Okay, two types of gene therapy. What are the two types of gene therapy? The first one is called gene augmentation therapy. First, I will take this gene augmentation therapy. Okay, I already told you therapy means treatment. Now actually in this treatment, there is a missing gene in the chromosome. There is a missing gene in the chromosome. For example, in the body cell, in the nucleus, I am taking one chromosome. Okay, now here one gene is there, next gene, like that. Many genes are present from end to end of the chromosome. Each gene is responsible for one particular character. Assume that the third gene is missing. We don't know where it has gone. Okay, now sometimes the radiation is passing through our body. The radiation is killing the gene, destroying the gene, so the gene will not be seen. So that is called the missing gene. Here one gene is there, already present, was present, but now it is missing, it is not there. Now what we have to do is, we should know, already we know the gene mapping and all that. What gene is missed? We have to find out what or which gene is missed. We have to find out. And the gene which is missed, we have to take from the other source and we have to introduce into this one. So what will happen? When the gene is missing, the character will be missing. So defect will be there. But once you bring the gene in the same place, same type of gene, again the character will be developed. So the individual will become normal individual. This is called one missing gene is there. In that place we have to bring a correct gene. That is called a corrective therapy. Correct gene we have to do. This is done by genetic engineers. It is the first therapy, gene augmentation therapy. The second one is gene inhibition. That is a treatment therapy. Gene inhibition therapy. Okay. Now, before I take this gene inhibition, you have to know the meaning of inhibition, suppressing, preventing or suppression, that is called inhibition. So something is a peculiar, we have to say. See, for certain characters in our body, okay, for every character, two genes are responsible. I think in the 9th and 10th Mendel's experiment or you might have studied. One character is controlled by two genes. The two genes are present in the paired chromosomes. For example, this is one chromosome, another one chromosome. It is common for all the plants, animals, everything. In this one gene is there. Here also the same type of gene will be there. Now these are paired genes present in the paired chromosome. They are responsible, both the genes are responsible for one character. Sometimes what will happen, this gene will be dominant gene. Even this gene will also be dominant gene. Sometimes this will be dominant, this will be recessive. Recessive means powerless, like that. So according to that, the character will be maintained. Now what I am going to do is, one example I want to give. Our brain. Our brain is very sensitive organ. Our brain is very sensitive organ. I am taking this example to understand the gene inhibition here. So the brain is very sensitive. The brain needs, okay, minerals, ions. The ions will be sodium ions, potassium ions and chloride ions. If these three types of ions are coming to brain normal quantity, the brain will be functioning normally. If these three types of ions are coming to the brain less quantity, nervous weakness will be there. Brain weakness. It is normal, but it cannot do the function actively. But another important thing is that if these three ions are supplied to the brain excessively, the brain will be damaged. So the three ions should not be supplied less, should not be supplied excess. Okay? So then the brain will be affected. Particularly excess quantity will stimulate, induce. The brain will be overexcited. I already told you it is very sensitive organ. Overexcited. As a result of which neurons will be bursting. If the neurons are bursting, brain failure will be there. That leads to the death of the individual. It is causing a disease called Huntington chorea, one major problem it is. So for this, how to avoid it, we have to see. So for the brain, for the supply of these ions in normal quantity, okay, normal quantities, a pair of genes are responsible, just like that. One is a dominant gene, D for dominant, one is a recessive gene. It should be like this, heterozygous condition. We are all normal, our brain is working normally. So in all our brain activities, in all of us, the genes are heterozygous condition. So for the supply of normal quantity of these three, only one dominant gene is needed. But what will happen for some people, the recessive gene is undergoing mutation, sudden change. So already one gene is there which is a dominant. Even the recessive gene will also be changed into dominant. When two dominant genes are there, oversupply of nutrients will be there to the brain. So the brain is excited, the brain neurons are damaged. So brain damage will be there. Now how to avoid this? When one gene, recessive gene, is changing into dominant, when it is both the genes are becoming dominant, it is harmful to the brain. But what we have to do is, one gene we have to suppress. Though it is a dominant, we have to suppress its activities. For that only, gene inhibition theory we are taking. What is that? Gene inhibition theory. So for the gene inhibition theory, what we are doing is that there is a gene in some individuals. So what gene is that? The gene is called antisense gene. Okay? There is generally some genes are antisense. What is the meaning of antisense? This gene is suppressing any other gene. That is called antisense gene. In our body, some places they are there, but normally they are not doing any function. But when it is needed, they will do it. What the scientists are doing by gene therapy, they are isolating this antisense gene. Now it is anti-suppressing character. Now they will introduce this antisense genes into the person where the mutation took place. Now what this antisense gene is doing? It is suppressing the action of, okay, suppressing the action of the dominant gene. Action of the dominant gene. So the dominant gene is not working anything. So only one dominant gene is there. So ion supply to the brain will be normal. So throughout the life, the individual will not have any problem. Even this is done by gene therapy. Okay, these are the two important treatments we are doing it. One for genetic defect, another one is for sometimes two dominant genes for a character may cause a harmful effect. So in that one dominant gene is suppressed, that is called a gene inhibition therapy. So two types of therapies we have studied. These two therapy or treatment are important in the field of medicine. It is also done by genetic engineers. Okay, two types of gene therapy. The first type of gene therapy is called somatic cell gene therapy. Okay, the first type of gene therapy is called somatic cell gene therapy. Now first we will take what is the meaning of somatic cell. Now therapy means treatment I told you. Now somatic cell gene therapy, we are going to do this type of therapy. That means they are altering the genes. Okay, altering the genes. That means fitting the correct genes in bone marrow cells, in blood cells, and also in skin cells, Now they are altering the defective gene into correct gene in bone marrow cells, blood cells, and skin cells. Now for this, understanding this somatic cell gene therapy. First, somatic cell. What is the meaning of it? Somatic cells means general body cells. Okay, general body cells. That means the body cells present in the skin, body cells present in the muscles, body cells present in the bones, body cells present in the blood, RBC, WBC, platelets, body cells present in the digestive system, respiratory system, circulatory system. Everything we call as general body cells. In the general body cells, if you are altering the genes, it is called a somatic cell gene therapy. In the body area. I want to take one example, that is skin cells. Say for example, one person is having normal skin, whether it is a medium color or black color or fair color, whatever it is, normal skin color is present. Now due to some harmful radiations, what is that? This is the hand of a person. Okay, normal skin color is there. But in one particular area, constantly the radiations are passing. For example, people who are working in X-ray department, people who are working in scan department, people who are working in nuclear power stations. Possibilities are there. Some harmful radiations are penetrating through the skin in a particular region for a long time, maybe 1 month, 2 months, 1 year, 2 years, 3 years, like that. Maybe after a period of time, in this skin, in this particular area, the skin color is showing a white patch. One white patch is there. It is a defect in the cells of the skin. The cells of the skin should produce normal skin color. The skin color is due to the melanin pigment present. So far they were doing, but now the cells are not producing the white patch. Sorry, the melanin. So the white patch is appearing. So what is the reason for this? In these cells, the genes are there for melanin production, but when the radiations are passing, the genes, normal genes for melanin are destroyed. They are destroyed. So what we have to do is, a normal gene for melanin, we have to isolate from the nearby region, area. And this gene should be introduced into one cell, and we have to make the cell to multiply. So slowly lot of cells will be produced, and the cells are spreading everywhere. These cells are producing normal skin. So slowly the white patch is disappearing. Okay, after gene is a normal gene is introduced, Now it is a disappearing, growing patch is a disappearing and normal skin color will be appeared. Now, why this is normal? How this normal skin color is coming? Because we are introducing one normal gene into one cell and we are stimulating the cell to multiply faster and faster so that the area will be covered. So this is called the somatic gene therapy. Now the somatic gene therapy, when we take it, it is non-heritable. Okay, it is curing only this particular area. So the skin color becomes normal. When the individual is growing, when the individual is having a child, this is a problem. Retouch will not affect the next generation. It is only for that particular individual. This is the first type of gene therapy, somatic cell gene therapy. Now we are coming to the second one, germline cell gene therapy. Okay, we are going to start with the germline cell gene therapy. Now that means the gene therapy, altering the gene. That means what? Substituting a normal gene in the place of defective gene. Is done in the germ cells. Germ cells are nothing but reproductive cells. Okay, now the reproductive cells, that is for example sperm for the male gamete, ovum is a female gamete. So each one is having nucleus, each one is having their own chromosomes and the genes. Now we have to alter the genes. There are certain genetic diseases which are hereditary, what we can say hereditary. This skin patch which appeared all of a sudden due to radiation is not hereditary. It is affecting only that particular individual. It is not hereditary. But here, lot of there is genetic diseases which are heritable, which are passing on to the subsequent generation. For curing that, the germline cell gene therapy is needed. For example, two parents are there. One parent is diabetic, which will go to the next generation. For example, father is a diabetic. That means what? The father is having defective gene. For insulin production, the father is having defective gene. Mother is normal. Okay, mother is normal. So here the normal gene is there. But in the sperm there is a defective gene. I'll put it like this. A defective gene is present. So what we have to do is, the defective gene should be completely removed. Okay, it is a defective gene. Then we have to take a normal gene. Okay, this is a normal gene. The normal gene should be introduced into the sperm. So this sperm is genetically modified sperm. In the sperm, no defective gene. Like all of the genes, even the insulin gene is also normal. Then the sperm, where the alteration is made, this sperm is allowed to fuse with the ovum. Zygote will be formed. So in the zygote, in the two chromosomes, both the genes will be normal for insulin. Because mother is bringing normal gene, now even the sperm from father is also bringing normal gene because nothing altered. Then the child is growing. Okay, the child is a normal child. The child will not be defective. For the child's father was defective. Father was diabetic, but the child is not at all diabetic. So the germline cell gene therapy is meant for, that is communicable, hereditary diseases. If male is affected, the defective gene in the sperm should be altered. If the female is affected, the defective gene in the female ovum should be altered. Like that we have to do this. This is called the germline cell gene therapy. So after that, when the child is going to adult, generation after generation if you take in the family, no diabetes mellitus at all. So the genetic disease which is inheriting from generation to generation is prevented by germline cell gene therapy. Thus we have completed two types of gene therapy.